give a brief overview on how to design primers for metabarcoding and how they are different from regular barcoding. And at the beginning, I prepare a longer presentation, so I have it in my computer. If you are interested, you can send me an email later, and I will send it to you. So the first thing that we have to remember when designing primers for metabarcoding is that although they have the same purpose as with regular barcoding, they do it in a different way, which is using many specimens and many species at a time. And that is going to change the requirements that we have for the design. So while with regular barcoding, we can use primers specific for some taxa or even species specific, and we are okay even when the amplification success is not super high, as long as we have enough material to do a Sanger sequencing reaction. With the primers for metabarcoding, we need to design them so they are universal within a specific taxonomy group, and that they have the same amplification success for all the species in that group, because otherwise we're going to find problems amplifying some species and sometimes even no amplification at all from certain species. So apart from technical requirements, primers for barcoding have to amplify a sequence that is variable enough to distinguish between closely related species. And it can be from 200-300 base pairs up to 900, for example. And the length of the primer is not the most important thing. It has to have a minimum 18 base pairs, but it can be up to 30. While for metabarcoding, the amplicon sequence must be variable enough to distinguish between species, the same as with barcoding, but it has to be franked by conserved regions within our taxonomic group of interest, so the primers can attach perfectly to all the species. And the length is limited, as in barcoding, in the short size for resolution capacity. Sometimes for very variable genes is enough with 100 base pairs, and in the upper limit is limited by the current sequencing technologies that cannot amplify or cannot sequence correctly with all the tags and the primers, and so more than 400 base pairs will be like a reasonable upper limit. And also when working with environmental samples, sometimes the DNA is going to be degraded, and we are not going to find longer fragments than this size. And the longer the primer is, the lower the probability that the entire region is conserved in our taxonomic group, so it has to be shorter. These are some examples of common metabarcoding markers for different groups. For bacteria is usually 16S. If we are aiming for all the eukaryotes in our sample, then we have to rely on 18S, and then if we are narrowing our interest, there are some alternatives for invertebrate CO1, for plants, different plastic markers, and most of them are mitochondrial, marked here with the M, and when they are not, or plastic, and when they are not mitochondrial, they are usually the ribosomal genes. So some things to keep in mind before choosing a marker is the copy number, because in a single cell we have many mitochondria and only one nucleus. And in the nucleus we have many copies of ribosomal genes, while usually there's only 2 copies of protein coding genes. Also degradation if we are working with environmental samples, because the mitochondria has an extra layer of protection and the genome is circular, so it's more protected towards degradation. The downside is that the mitochondrial genome has much higher content in A and T and lower in CG, and that difficults the design. Also, we have to consider if it's going to be ribosomal or protein coding genes, because the protein coding genes are super variable in the third position of the codon because of the redundancy of the genetic code. And the ribosomal, they are not that variable, but they are very variable in length, and that can also mean problems during the sequencing later if we have longer fragments or shorter that are more sequenced. And also, if we are aiming for getting a species list, or we are okay just with diversity indices, and we are fine with OTUs, because if we are aiming for a species list, we have to choose a marker that has a comprehensive reference database. But if we are only interested in number of OTUs, then we can design our own custom markers. So we have this is a plot of different genes. This is the diversity of sequencing and alignment of around 2,000 insect species. And at the beginning I said we have to find primers that markers that are very variable in a region, but very conserved in the regions surrounding them. So there is a trade-off between resolution and the coverage of the primers. So we have very high mutation rate genes that are almost impossible to find good regions for primer design because they are not very conserved. So these genes are not very useful for us. Then we have high mutation rates, for example, CO1 or cytochrome B, that are very variable as well, but they have some regions with lower diversity, and then it's in these regions where we have to look for good sequences to design our primer. And then lower mutation rate genes, in this case the ribosomal ones, that have much more regions that are conserved, but at the cost of not being that variable. So in this case, it's good to have primer design, but then the amplicon should be longer to have enough resolution. Yeah, this is what I mentioned before. So this is the ideal situation for a primer design. Let's assume that we have an alignment and we find a very variable region flanked by a very conserved region. In this case, there will be very good primers because they don't have homopolymers, the GC content is high, evenly distributed, and with Cs and Gs in the 3' end that they are going to form triple hydrogen bonds in contrast to the AT that are going to form only 2, but this is never going to happen. We're never going to find super conserved, wonderful regions surrounding very variable. What is more likely to happen is that there is going to be a most abundant sequence that we can say, okay, let's use this sequence as our primer. But then if you look in the alignment, we find that some species in this position have an A, some others have a T, in this one some have a G, some a C, and it's never going to be the one with the G, is going to have an A, and the one with the C is going to have a T. It's going to be all the different possibilities that are going to difficult your life. But let's assume that we choose this sequence as our primer. Because of these differences here, these mismatches, what is going to happen in the PCR is that some species are going to have more affinity to our primer than others. And then during the amplification, we were talking before about quantification or doing a quantitative approach. This is going to destroy our hopes of doing that. And in the worst case, we're going to find that some species are not being amplified at all. So one easy way we have to solve this problem is using degenerate primers. Degenerate primers are primers that have some letters that are like a code for various nucleotides. So it means that when we have this sequence here, a B is not a nucleotide, but is indicating us that in this position we are going to have in some primers a C, in some others a G, and some others a T. So at the end, what we are going to have in our vial that we get from the commercial house is a mix of, in this case, 12 sequences. And there are degenerate bases that stand for 2 nucleotides, some others for 3, and then the N for 4. And in this case, this will solve this problem because some primers will attach to their perfect match, and then we will have a more even amplification. This sounds like the perfect solution for everything, but too much of something good can be bad. We have to be careful in not designing primers with too high degeneracy. For example, I think for protein coding genes, I've been using up to 200, degeneracy of 200. That means that in my vial, I have 200 unique primer sequences. And some of them are going to be needed, some of them are not going to be needed. But the risk that it has is that since they can be very variable, sometimes they can bind to different sites in the genome that we are not interested. They can bind to other taxa that we are not interested at all. And for example, with environmental samples, if we are doing mitochondrial ribosomal, they are homologous to the bacterial ribosomal, so we might find amplification of bacteria when we are looking for, I don't know, microarthropods, for example. And also, with each new unique sequence, the concentration of each one is going to be lower. And then, for example, in this one, we have that there are more sequences with a T here and less with a C and J, or G, sorry. But in our mix, there's going to be 1/3 of each. So the proportion of sequences in our vial is not going to match the proportion of sequences of the template. So we have to be careful when introducing degeneracy. Finally, I think also depending on how much sequences we have for the primer design, we can use, I classified them in 3 different packages. So the non-computational is bioedit, for example, or Genius. You get your sequences, you have like 100, 150 or something like that. You do your alignment, and then these programs are going to tell you the consensus sequences and where the conserved regions are, and so. When you have a little bit more of sequences, then you can use what I call visual computational. In this case, the program Primer Miner is quite useful. It's an R package. It helps you downloading sequences from GenBank and Bold, but you have to do your own custom alignment. And this program will help you indicating with colors where you have more sequence diversity or where you have more conserved regions. But it's yourself who has to design the primers. And finally, the computational, when you have thousands of sequences, is the most useful way. And there are 2 programs, Eco Primers, that is, well, you do your downloading of sequences, but then it's alignment free. You just enter one command and you will get a list of primers. It's part of the, I don't know if you know, Obitool package is package designed for metabarcoding bioinformatics. Yeah, so it's quite useful. And also DG Prime, that is a little bit more, I don't know, you can play a little bit more. You have to download your own sequences, do your own alignment, but then the program will look for good regions for design primers. And the good thing is that it will suggest you different primers, the generate primers, which is something that the rest of the programs don't include. Esto no es... Okay, so I changed this morning the presentation and I thought it was here. Once you have designed your primers, before going to the lab and ordering them, it's good to do what is called an in silico PCR. And it's a program that simulates the amplification given a set of sequences that you have and the primer that you have designed. And there are different indices that you can use to evaluate how good your primer is. And those are taxonomic coverage, which is just a proportion of the species amplified versus the total species of your database. Taxonomic or exclusive taxonomic resolution, which is the number of species identified from those amplified, and then an index that is the proportion of species identified from the total species that you had. It's very important when we are doing the taxonomic resolution index to think what is it? I'm okay? Okay. To think what we are going to consider as an ambiguity. Because, for example, with the index BS, we have here, let's say that we do the in silico PCR and then we do a clustering of the sequences that we have obtained and a taxonomic assignment. And we find these 5 MOTUs. We see that there is only one MOTU that has taxonomic disagreement, because some sequences are considered as Bombyx-Murray and some others as a butterfly. So we say, okay, we have 5 MOTUs and only one of them is not a good taxon. So yeah, this primer or yeah, this marker is super good. But as we see here, we have Drosophila simulans and Drosophila simulans. So what this marker is telling us is these 2 things that are the same are actually different, and that is incorrect. So we have to be careful to consider these MOTUs as ambiguities, and only those that don't have any taxonomic disagreement could be good. So in this case, this marker will not be optimal. That's it. Thank you very much for listening. As I said, if you want to see the long presentation and you have questions after the workshop, that's my email.
Thank you, Daniel, for your explanation of primer design. Do you have any questions regarding?
About that in silico PCR, I never heard of it, so you could elaborate.
Yeah, there are some programs that will. Also with the OviTools package, there is together with EcoPrimers, there is EcoPCR, and it's command line programs that will look at your reference database with the primer sequence that you have introduced, and they will basically give you an output of the amplicon that you are looking for. You can say, I want, with this forward primer and this reverse primer, I want an amplicon that is from 50 to 500 base pairs.
But do you fiddle around with parameters like the annealing temperature?
temperature?
No, not with annealing temperature.
So it's just about the primers?
Yeah, it's...
Should,
Then you can choose, for example, how many mismatches are you going to allow. If you want those mismatches to be not in the 3' end. Yeah, there are some parameters that you can use, but it's never going to be as real as a true PCR. So after you have decided that you have a good candidate as a primer, then you have to order them and do in vitro testings before, yeah.
So you're working with insects?
Yep.
Which marker would you use as a DNA metabarcode, CO1 or the 16S?
16S. Well, I will use both together because the problem with most CO1 primers is that they neglect Hymenoptera, wasps, bees, and ants. Most of the primers that work almost perfectly with, actually the primer that I have found that works better with all the rest of the insect orders, they amplify like 40% of the Hymenoptera that I had in the database. But then 16S was very good with Hymenoptera and other groups. And the resolution, the taxonomic resolution was as good as with CO1, but with a longer fragment.
And regarding the very last slide, we had that
Yep.
exactly. You were saying that that index reveals that those primers are not particularly good because they cluster the same species into two different OTUs, right?
Yep.
Can't they be reclustered after being made? Like once you have your list of OTUs, can't you just run some sort of program to cluster those that are identical? I mean,
Yes.
the same last.
Yes.
I don't know if that's.
Yeah, actually, there is a very, I think it's from January this year. It looks into the co-occurrence in the same sample of clusters with the same taxonomic assignment and decides if it's an error or it's, yeah, you can do it. This is basically more important when you are not looking for a species list. Because if you're looking for a species list, you say, okay, I have Drosophila simulans, I have a second OTU that is Drosophila simulans, but I don't care because I already know that I have Drosophila there. It's more important when you are doing, I don't know, richness and you are looking into OTUs instead of a species list or doing diversity indices.
So these analyses, do they contemplate for variability within the same species? I mean,
yes.
The barcode is a bit diverse within Drosophila simulans, so.
Yeah, you can. There are programs for clustering that have a variable threshold. So you set a maximum, and then it will start looking from 0 to, let's say that you say, I have this 320 base pair long CO1, and I know that the barcoding gap is at the 3-4%. So you say, okay, I'm going to allow 12 nucleotides of difference to consider it as the same species. And then it will start looking to those that have only one, and then creating a second layer with the ones that have two. Yeah.
One question. Because you work in a natural history museum and with insects, do you work with any type of sample like amber fossil or
No.
DNA or something?
No, I've worked with malaise traps that collect all the insects directly in a jar of alcohol, so it's very convenient because they sample themselves, and with soil samples. Yeah.
There's not any other question. We have a little bit delay, but not much, so we have 30 minutes to have a coffee break outside there.
you.